Hello there, Dr. DiMaio here. Yes, we're doing the innate non-specific immunity. We have a PowerPoint for our course associated with the immune system that was too big and too broad to try to do a recording. I tried 3 times to record it, and because of the amount of content, it wouldn't let me record it. So I'm breaking it down to bite-sized pieces, and it helps to section it off and then review what you just covered after we do this. So this is the innate or non-specific immunity. Basically, you can look at this in different ways, but the 3 types of immunity we will discuss throughout these different lectures. This is the first one we're going to be doing on the innate immunity. That's non-specific immunity, meaning that it does not need any B cells or T cells for this to operate. And so basically it covers several different things, and we'll talk about them in a moment. And you have a first line of defense, a second line of defense, and then I add this third line of defense, which is really like a bridge between the innate and the adaptive immunity. So what the heck is the adaptive immunity? Adaptive immunity has 2 parts: cell mediated, that means it's cells attacking cells, and then there's the humoral, and the T cells are associated with the cell mediated, and the B cells are associated with the humoral, and yet they all work together. And then there's something called passive immunity. I added this as a third type. I mean, really is not a third type. It's just something that occurs passively from mother to fetus. The mother's has immunoglobulins that she passes directly right through the placenta, and then also in breast milk, it can actually help to pass through passively to the baby. As an adult, we could get immunoglobulin injections. I watched just yesterday Dr. Rand talk about how nobody discussed this, that you could take an immunoglobulin from someone who had already had the infection and inject it into healthy people to protect them and give them the immunity. And basically what he has discussed, and he has been, since he is a eye surgeon, he's a very brilliant man, and he has said, when you have had this infection, this viral infection, the COVID-19 I'm talking about, you have immunity. We don't know everything about it because it's new, but generally speaking, we're discussing general anatomy and physiology, and in general anatomy and physiology, we produce memory cells. Those memory cells are produced from this part, the adaptive immunity. The adaptive immunity, or specific, has to do with the production of T cells and B cells, and then antibodies, and then we have memory cells for all of that are produced. Whenever you're making these T cells and B cells that are specific for a specific target antigen, we go through this thing called clonal selection, and we make clones of them, and then we make memory cells. It's a pattern that happens. Every time we have an immune response to something, we create these new T cells that are adaptive to try to attack that specific antigen. And then we also make specific B cells to make a specific antibody for that specific antigen. At the same time, they make clones, many of them, and they make memory cells. And we have these memory cells that if you tested somebody for the memory cells, you won't find them unless that person's been exposed to that same virus again. So the memory cells are dormant. They're not circulating through your system. You can't draw them from your blood and say, oh, he's got the memory cells. No, the memory cells are in your body, and they will come out. They start making new ones when you're exposed to it. This is something that people don't generally know. Okay, so that's a pretty big nutshell on that one topic. So we're going to start talking about this innate immune system. The innate means it's non-specific, and it has to do with some lines of defense. The first 2 lines of defense are the big ones we usually talk about. And the first line of defense is, let's think about it. If something's going to try to attack you, you've got like a shield around you, and that would be your skin and epidermis with these keratinocytes. And by shedding your keratinocytes, the fact that they do shed these dead cells, you're actually removing microbes. They're leaving your body. that would on your skin. Then you have mucous membranes, and it's got mucoid material, which is like a viscous material, which that in itself helps to trap things, and the hairs help to trap things, and then that helps to decrease the amount of microbe growth. And then some of our fluids are designed to protect us, like tears, saliva, urine, vaginal secretions, and when we defecate or vomit, we are reducing the microbe growth in our body. All those things do that. There's different pHs of these different things. And when we look at the oils or sebum on our skin and hair, it has a pH of 3.5, which is quite acidic, which protects from growth of microbes. So you see these dots, it's growth of microbes, protecting from growth of microbes, protecting from growth of microbes, protecting from growth of microbes, and even your gastric juice is extremely acidic. We know that in the stomach it's hydrochloric acid. That will it'll burn a hole in your car. So that will definitely have a hampering on the growth of a microbe. Our second line of defense is these substances that are found in our body or in our plasma and in between cells. So you have these internal microbial substances, such as interferon. This is a really cool one. It's produced by infected cells. We'll talk about that. And then we have the complement system, which is made up of 35 different proteins that are produced by the liver, but they're circulating and they're inactive until they become activated. When they're activated, they actually really enhance the immune response. And then we have these iron-binding proteins just found in our body associated with lactoferrin or ferritin, and it helps decrease the growth of microbes. And we have these antimicrobial proteins, very specific proteins associated with the immune system. Let's get to it. Let's get to these different things. So let's talk about this third line of defense first, and that there is, so to speak, a third line of defense of the innate immune system. We're talking about things that are innate, that just are there, ready to protect you. And we have these other cells lying in those tissues that we just talked about within the mucous membranes and just subcutaneous tissues. We have natural killer cells and phagocytes. We'll talk about Langerhans cells later, which become mobilized and go through another step for the adaptive immune system. And these cells are functional without specificity, and then they can help the specificity. So they kind of are a bridge or a gap between the innate, non-specific, and then the adaptive. And this is a wonderful thing we have in our body. Okay, so let's just talk about the innate or non-specific internal antimicrobial substances. And one of the proteins is an interferon. Interferon generally stops the replication of virus. It's produced by a cell that was infected with a virus. So it functions a lot like a paracrine in that the infected cell becomes the paracrine cell now and produces interferon. That's after it became infected. So maybe there's no hope for that cell to survive, but he's going to try and save all his brothers. So whenever any cell in your body becomes infected with a virus, it will now produce this chemical called interferon. And interferon is a protein. Now that interferon will leave the cell that was infected and then bind to an adjacent uninfected cell. Doesn't matter what cell is next to it, it will bind to them and protect them from the virus and stop the replication of the virus in its tracks. So now if I was sitting next to you as a cell and my brother was sitting on the other side of you as a cell and you got infected, you would produce this interferon. It would come to our membranes. We would now incorporate it and would help to protect us from that viral infection and stop any virus from replicating as well. Isn't that wonderful? That's a wonderful thing. This is just naturally found in our body. And then we have the second part of these chemicals, and these are proteins called complement, and they're part of what's called the complement system. It's actually over 30 proteins produced by the liver, circulating in the plasma and your body tissues. These proteins will have this cascade occurrence. Now, they're inactive, but they'll become activated. So the protein cascade it begins with 3 different possible pathways for them to be activated. All the 3 pathways will end in the activation of C3. That's one of the complement proteins. Remember I said there's over 30 of them. So there's the classical pathway, the alternative pathway, and the lectin pathway. The classical pathway is when there's an antigen-antibody complex. Now, that antigen-antibody complex had to come from a B cell producing an antibody, and that was from the adaptive immune system. All right, so let's say this antigen-antibody complex is floating around, and then it binds to C1, and eventually it will bind to C3. And C3 becomes activated, and those fragments of the C3 initiate 3 very important things you can imagine: phagocytosis, cytolysis, and inflammation, all necessary for a healing response. We'll talk about that in a few moments. The alternative pathway does not involve antibodies. Is that clear enough? The alternative pathway does not include antibodies. That means there's nothing to do with B cells. The pathway is initiated by the interaction between these complexes that are on the antigen, lipid carbohydrate complexes on the surface of the microbe, and complement proteins, factors B, D, and P. And then they activate again C3 and will trigger these things up here: phagocytosis, cytolysis, and inflammation. The lectin pathway is macrophages release chemicals after ingesting a microbe, and these chemicals trigger the liver to produce lectins. The lectins bind to the membrane carbohydrates of the microbes, causing activation of C3, which would then lead to phagocytosis, cytolysis, and inflammation. So here, again, we're talking about complement. And this here shows you a chart. It shows you the classical pathway with the antigen-antibody complex. Shows you the lectin pathway with the lectin binding to the pathogen surface. Shows you the alternative pathway with the pathogen surface. And then you get complement activation, which will create phagocytosis. Now, this term opsonization is how it helps phagocytosis. So opsonization is how complement actually helps phagocytosis. It prepares it, helps it to bind better to the membrane of the microbe. So opsonization is an enhancement of phagocytosis. And we'll talk about phagocytosis in a little more detail, and I'm sure you all know about it a little bit. And then cytolysis, this is really cool. What happens is once that C3 becomes activated, it creates more proteins that come to that membrane, and those proteins form what's called a membrane attack complex. We're going to talk about that in a moment, which creates a perforin. A perforin is like an ion channel protein that penetrates the membrane of the invading microbe, like putting a tube in. And I talk about the doo-doo. We'll talk about that doo-doo in a moment. And then some of those fragments are all around there, so that triggers inflammation. So let's look. So all 3 complement pathways lead to activation of C3. After C3 is activated, it leads to the following 3 events. We said phagocytosis, opsonization, and phagocytosis. The C3 binds to the surface of the microbe. The macrophage receptors now bind to the C3b. Thus, opsonization promotes the attachment of the phagocyte. So the C3b binding promotes this opsonization, and the opsonization promotes the phagocyte to the microbe, enhancing phagocytosis. Cytolysis is when the C3b initiates a cascade and results in cytolysis. The result is this formation of a membrane attack complex, the MAC. Yeah, baby. We want a MAC form fast. And C3b splits C5 and C5b, fragment binds to C6 and C7. And you can imagine all these proteins coming and accumulating and forming this cylinder. They literally form a cylinder-shaped protein ion channel called the MAC, membrane attack complex, or a deuterium. You know what a deuterium is. If you had me in A&P1, you know. I think I mentioned it once this semester. This is the summer of 2021. I had mentioned, do you know what a deuterium is? And I said I use the term Italians because that's what I remember as a kid. It was a silly joke. Hey, kid, you want something? I got a deuterium for you. What's that? And the guy gets the end of the paper towel roll and he puts it to his mouth. He goes, doo doo doo, and he gives it to the kid, and he's running around the house going, doo doo doo, with the paper towel roll. The cardboard, the end of the paper towel roll. So it's like a tube. So imagine if you take a cell membrane, imagine that cell membrane. Now let's look at an analogy of a cell membrane as being a box, like a refrigerator box. And you cut a hole just wide enough for the doo 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 to pass through. You place the doo-doo-doo in, and now you can put a hose in and flood your brother inside his box there. So that's basically what happens when you put this ion channel in. You're surrounded, the cell is surrounded three-dimensionally in fluids, and it causes fluids, the extracellular fluids or the fluids outside the cell, to rush in and cause the microbe to burst. That's called lyse. So cytolysis. And then all these fragments are all over the place, and they trigger C3a and C5 to bind to mast cells and trigger the release of histamine. Histamine, we know, promotes blood vessel dilation or permeability and inflammation. It also, this C5 will attract the phagocytes through chemotaxis, and that's the chemical to attract them. Now we have other types of proteins that are antimicrobial. And let's talk about iron-binding proteins. They inhibit the growth of certain bacteria by reducing the amount of available iron. So like ferritin will bind to iron, as will hemoglobin. This is giving you an example of lactoferrin. And then there's another one called transferrin. These are all proteins that can bind to iron and reduce the available iron so that there cannot be a growth of certain bacteria. They require iron. Then we have innate or non-specific antimicrobial proteins. These are specific AMPs, including dermacidin, defensins, thrombocytins, and featured here in this picture here are the cathelicidins. Human cationic antibacterial protein, or also called LL37, is the only cathelicidin in humans and is primarily found in the secondary granules of neutrophils. Remember, a neutrophil is a granulocyte, and the LL37 is released as an active domain from macrophages, monocytes as well, and even epithelial cells. So in addition to killing a wide array of microbes, AMPs can attract what's called dendritic cells. Okay, we're introducing this term dendritic cells, and also can attract mast cells. So we'll talk about what a dendritic cell is, and just quickly, just so you understand, they're a specific type of cell that can become an APC. That's an antigen presenting cell. And if you remember, we learned about the skin, we learned about the islets, and we learned about Langerhans cells under the skin. Do you remember that? And when I teach, I said that's a white blood cell right underneath your skin. You're not even in the connective tissue proper yet; it's just below it. And so they can be. That's one example of the dendritic cell. A macrophage can become a dendritic cell. So a dendritic cell the idea is that it's going to have a complex on its surface that's going to take from the microbe, and that complex is going to be like a flag identifying those bad guys that have infected the body. So the good news here about these proteins we're talking about here, the defensins, the thrombocytins, is that microbes cannot develop a resistance to them. So you want these natural chemicals in your body, which your body produces. And so these are proteins. Now, what is a protein made up of? Proteins are made up of amino acids. And here's another example as to where I always talk about the 8 essential amino acids are important for your body to function well. Let's say you were a person who had a diet that consisted of the same proteins all the time and never had a diversity of proteins, and you were missing, let's say, a couple of amino acids, which are essential. There are 8 essential amino acids for life. This is one example where if you're not getting those 8 essential amino acids, you won't be able to produce these proteins, which are wonderful. Now, when we talk about antibodies, they're made up of 400 amino acids and 250 amino acids. We talk about these antibodies, and the shape are made up of these amino acids. If you're not getting enough of those 8 essential amino acids, you could quite possibly be diminishing your immune system, and nobody would really know it. So nutritionally, you can actually affect your health in a negative way or in a positive way. And it goes even further with those essential amino acids, but we won't even get into that right now. Okay, so the third line of defense, of innate defense, possibly a bridge to the adaptive immunity, is when microbes penetrate your skin. Now they went below the mucous membranes or the skin, and they're bypassing all these different microbial substances we just talked about. They survived it somehow, and even the intestinal fluids and the blood. The next non-specific innate immune response is accomplished via natural killer cells and phagocytes. So natural killer cells make up about 5 to 10% of all lymphocytes, and they're found in your lymph tissues such as your spleen, your lymph nodes, your red bone marrow, and they have the ability to attack any cell that's really not one of us. What do I mean by that? The cells have on their surface a membrane proteins. So any cell that doesn't have a usual or a normal membrane protein would be identified by the natural killer cell, like a guard duty. Halt, who goes there? Let me see your ID. They have the wrong ID, even including cancer cells will. So they can attack it right away. And that's very important. And they do attack it very similar to what we just discussed with the perforins. So the natural killer cell will bind to the target cell's MHC, major histocompatibility. And if you don't know what major histocompatibility is, that's your cell identity marker. So on the cell membrane of every cell in your body, you have these glycoproteins that tell you that you're you. That's who you are. And like if you're going to try to do a tissue transplant, they try to identify your cell identity marker and find one that matches the best. So nobody is exactly the same. So the natural killer cell will bind to these abnormal or foreign MHC complexes on top of those microbes and then release toxic granules, chemicals, and they do it by creating a protein ion channel called a perforin, and they inject it basically, and it causes that cell to explode or cytolysis. You have these things called granzymes, and they also produce cell apoptosis, killing the infected cell and microbes. So if it's an infected cell, they actually will destroy that as well. Now, when it destroys that, it's taking some of that protein with it, and then that can be now an APC as well. Now he can be an antigen presenting cell that we're going to talk about when we get into the adaptive immune systems. That's why I say this is like a bridge to the adaptive immunity. It's happening automatically. It doesn't need any specific B cells or T cells or anything like that. But once he engulfs that and takes a piece of that, now this natural killer cell can become an APC as well. All right, so let's talk about phagocytosis for a minute. There's basically 2 types. This is very general and very quick. Neutrophils are phagocytes, but they're macrophages. They're not big. And macrophages are large. And so macrophages could be fixed or wandering. A common macrophage would be a monocyte. But also eosinophils can be microphages and macrophages. Phagocytosis has 5 phases: chemotaxis, adherence, ingestion, digestion, and killing. So chemotaxis, we know the chemicals released by damaged cells. microbes, white blood cells are activated complement will attract the phagocyte, macrophage, or microphage to the location. Then adherence is when the attachment to the invader, the complement, will enhance this. So complement proteins can help the attachment to that invader, which will help in phagocytosis. And then when you phagocytose, you're ingesting this thing, forming it into a phagosome within the cell, and then digestion through enzymes, breaking it down, and it causes the burst and destruction of the microbe. This may leave many residual bodies that need to be removed after you have this death of this microbe. Here it is again. So here's a phagocyte, and here's some bacteria with these chemicals around it, attracting it towards it. It causes pseudopods and brings it in, forms a phagosome, and the phagosome forms a phagolysosome, and has enzymes to breaks it up into its individual products and releases the cellular debris.